the way, all the audio we need today is just me screaming into the room and maybe you asking questions. That makes the setup apparently much faster. So in theory, we're good to go. If you have any questions or ideas, concerns from last time, I'm leaving behind this hidden messages and motive search. It was a bit rushed at the end last time about these random search heuristics. The thing is, there's not so much you can really say about them other than showing specific examples on certain data where they seem to work well. And then the last one, this Gibbs sampler, seems to have an edge there. But this, I mean, people would love to be able to prove something. And I think this is just in general seems hard, and not much is known. So not much I can add to what I said last time. But I think these heuristics are generic enough that having seen the idea behind them might be useful. Even if we can't necessarily say much about why they work or how well they work. Any questions, concerns? If not, then we'll make a pivot to a more algorithmic topic. Maybe you can also close the door, Tommy, when you come back. Might be nicer sound-wise. And if someone comes late, they can open it again. In the next unit, we'll look at some algorithms to compare sequences. This is in hidden versions. We've looked at this before, but let's do a more systematic study of what can and cannot be easily done there, and become also, let's get a bit more quantitative and precise what really we mean by comparing sequences or similar sequences and all that. And the chapter is a lot more algorithmic in a way, with clean and kind of basic algorithms where you can say something about. The tentative outline has a lot of topics I'd love to say a little bit about. They start from extremely basic and go to much more sophisticated. So maybe the level of coverage will have to be adapted to that a bit. Let's start with the basic motivation first. As always in this module, there's some biology behind it. I've shown this picture at the very beginning as one example of a protein that fulfills an important function. That's hemoglobin, or the human hemoglobin to be precise. And you see it consists of these 4 colored things. Now, each of the colored strands is one protein chain that folds into this specific 3-dimensional shape. And there's actually 2 that are always equal. The blue ones, they're separate, but they're identical copies of the same protein. Same for the red one. And they have these names, the human hemoglobin subunit, either the alpha version or the beta version. The green bits that are sticking in there, which in this picture maybe is a bit less easy to see. Conceptually, there's these 4 blobs that connect, and each of them has a little, this green part, a heme group, which is a non-protein molecule with 1 iron in the middle. So there's 1 iron. So each of these hemoglobin complexes can capture 4 oxygen molecules, 1 in each of these pockets. They attach to the iron and then, well, can release again, depending on the chemistry of the environment. So this is really, really small. You have lots of those molecules in your blood cells. And what you can maybe guess from the schematic drawing, and if you rotate this thing in the right way, also see in the 3-dimensional structure, is that the red and the blue proteins are very similar. they're essentially fulfilling the same kind of function. And in particular, they have these parts that hold the green parts in place, which are highly conserved. And if you modify them, then they don't work anymore. And usually people don't survive that. So all living beings that have blood cells of some sort will have very similar proteins here. And let's take this as an example. Now, it seems evolutionarily why we have the alpha and beta version. As always in biology, there's more varieties, and then there's specific diseases that also express slightly different proteins. It might just be an evolutionary coincidence that these two, they certainly come from a common ancestor. Then at some point there was a mutation and they branched off. And then by forming this structure where the opposite ones are the same, maybe they even evolved to be a little more stable that way. But from my understanding of the biology, it's not clear that you would need these two separate copies. You could have maybe lived with 4 identical versions of the protein. But that's what it is. Now, at least the claim is these alpha and beta globin versions should be very similar. Now you can click on those links. That's a database with all the proteins that are known. And you can look up the amino acid sequence of these 2 proteins. That's the red one and the blue one. They're not exactly the same length. That's the first nuisance. And I know it's fairly small, and I had to squeeze it together to fit them in one line. Maybe you at least visually agree that they don't actually look all that similar, and that they fulfill the essentially identical role is no longer visible. There's 2 reasons for that. One is that if you stare at it for long enough, you may see that there's some parts like this KKV, but then AL. So it's not like there's nothing to be found that is similar, but it's shifted. There's a second reason, which is more annoying because I didn't give you a chance to see it. There's some amino acids that are replaced from one to the other, but their chemical function and structure is very similar, so that in fact they can fulfill the same role, but as a letter they are different. And so our abstraction to represent them by some arbitrary letters in the alphabet is not so helpful here. But the second part, this first part, that there seems to be similarity, but it is not in the same places, means you have to at least work a little harder to find it. And that's what this unit is about. These don't look so similar, but there's tools that specifically also know that these are proteins, and they form what's called an alignment. So you strategically stretch either of the strands by inserting these gap symbols. So you just make room for the other strand to catch up. And you see this vertical line in all the places where there's a matching character in top and bottom. And in addition, there's these colons, which means there's not the same character, but they're at least chemically similar. So if you add those 2 up, there's 65 of these exact correspondences and 25 more where the amino acids are at least similar. The 2 are indeed 60% the same, and 60% the same structure or function. Then there's a few places where there's been stretches or deletions, and there's a few places where amino acids have been replaced. My hypothesis would be that if you trace this to where the structure is, that they may be just not part of this relevant bit in the middle that keep the heme group in place, but they are in a position where it doesn't matter. And that teaches you 2 things. Like, number one, you can definitely use this to find highly similar things. 60% the same. That's for biological sequences, that's like identical. This is amazingly good match. Then the second thing is, by forming this alignment, you learn a lot about the different parts. You learn the highly conserved parts, Probably that there's these 4 identical matches separated by 1 and something more. Maybe this is the central part around the green thing. So you can identify the highly conserved regions locally. And you might also have a hint, maybe this one, this long stretch that got inserted, you can locate and try to find or combine knowledge from different pieces of evidence that you have. And this is another piece of evidence. It tells you which strands may be less important because they're more variable. That's the kind of backdrop. Now we'll look at how to compute those and how to modify the basic algorithms to various needs that are slightly similar to this one, but have some other features. From computer science perspective, we're essentially talking about string distances. So we have strings that are not the same, but also not completely random. And we want to measure how close they are in some well-defined way. In bioinformatics, this always comes from mutations, but we'll abstract from that most of this unit. We'll look at various options, but the good thing is that they are algorithmically somewhat similar. So we often can do a unified framework for solving them. But there's a few also algorithmic tweaks that you can apply to these kind of algorithms that maybe are not so obvious or are, say, adequate for a master's level module. The last statement we'll only come back to at the very end. The general problem, if you're looking for these alignments, is a hard problem when the number of sequences grows. For 2 sequences, you'll see life is fairly easy. And we have efficient algorithms that find best possible alignments in the sense we will define. If you add more sequences, then this becomes much harder. And this is unfortunate because oftentimes what biologists would like to do is take the same protein, they know the gene in the DNA, then compare that gene across different species that are more or less closely related or have diverged from a common ancestor more or less recently, and compare which parts are conserved, which are variable, all these things. And then you want to do this with a whole lot of sequences, dozens or hundreds. And this is still not solved. However, the underlying problem is NP-hard, so it's going to be tricky. But I think this is still actively worked on how to best deal with it. Okay. We've already seen string distances, the Hamming distance, but it has a major shortcoming. I mean, Hamming distance was just defined as the number of mismatches, and it was only defined for strings of the same length. So there was no question of shifting things around. So you can't even use it if the strings are not the same length as in our protein example, but more critically, if you have strings like these two, which are largely identical, if only you were able to notice the small shift. Hamming distance would still say they're maximally different. That's not useful in our context. So clearly Hamming distance is not the one. The good thing about Hamming distance, it's trivial to compute. You just scan the strings and that's it. There's no optimization process behind it. Okay, what can we do that's a little better? I guess most natural definition, ignoring all of biology for now, is edit distance, where you ask yourself the question, assume I start with one string and I'm allowed certain small operations that modify individual characters. How can I get from that first string to the second with the smallest number of operations? And as a concrete example for these 2 strings, I mean, I claimed these should be very similar because they have a large shared substring. So we could take the first string, I guess that should be A, the way I called it above. I'm deleting the first character, so that one's gone. And then I'm inserting an S at the end, and then the claim is I arrive at B. And you can verify in this case. So here the edit distance is 2. It's at most 2 from this edit script, which proves I can do it with 2 operations. Now you would also have to prove for the equality that you cannot do it with 1. But since there are 2 positions that differ, I think this is kind of evident by just trying all possible operations. A single one will not fix both places. Which also means that this edit script is an optimal edit script because it has the fewest operations among all that work for A and B. The general algorithmic problem then is given 2 strings and will always have A of length m and B of length n. So try to keep that in mind. I'll try to repeat it on the slides wherever it's needed. In general, they are over some alphabet. Most of the time, it will be for biological sequences, either 4 letters or 20 letters. So in particular, the alphabet most of the time is fixed and is not growing. Makes some of the algorithms easier. Okay. We'll focus in the algorithms on computing the distance itself as a number, as opposed to outputting the edit script as a kind of procedure to get from one string to the other. We'll come back to also producing the script. The point here is all the algorithms we'll look at, all the algorithms I'll present in detail it's very easy to augment them from just computing the distance to also producing the edit script. So it's not a hard step, but it makes the formulation a bit more concise. Okay. Let's see another small example, algorithm logarithm. Here's a concrete edit script, similar to the previous example. I want to connect this explicitly to the biological motivation, where I didn't talk about edit scripts, although I guess I could have. But for this quick visual information about the 2 sequences, this alignment, this writing of the 2 strings on top of each other with some annotation in between potentially, that's more convenient. And the 2 are really equivalent. That's what I'm trying to convince you of here. Whenever you have a deletion, then you'll have a character where there's a gap symbol in the corresponding second string. Whenever you have an insertion, like here in that position, you have a gap symbol in the top string and the letter in the lower one. And the other option is a replacement of a character. I'm taking O and turn it into an A. Then these are just put on top of each other, and I'll have to kind of count this as a mismatch. Whereas for the other characters where they're put on top of each other, I'm counting them as a match. So you can easily go from edit scripts to this alignment notation and back. It's kind of equivalent ways to represent the information. The edit script has the benefit that its size is bounded in terms of the edit distance. And the string alignment, because you have to read the strings, you could produce it in the same time. Sometimes it's more convenient to think in terms of one, sometimes more in the other. So should not get confused by language switching back and forth. For the algorithms, it's sometimes convenient to represent the alignments a bit more formally, so I'll just write them as these pairs of characters. The middle line is not represented because that's implicit. I can have a match column, so that's the same character in top and bottom. Or I can have a deletion or an insertion. That's one where I have a gap in one of the strings. And I can have a replacement. And so the alignment formally for us is a string over this alphabet of pairs. Okay. We'll write it in this more intuitive way, but if you have to properly define things, then it's a string over this pairs of characters. The only reason I wrote this in this more complicated way is we don't allow gap as a pair, right? This would not make sense. No optimal edit script will ever contain this. So let's not even include it. Okay, if you have an alignment written like this, then obviously counting the red things is giving you the edit distance. I'm saying this is all very, very basic stuff, but I don't want to rush it over in case you've not seen edit distance. I don't know if it's a lot of details to take in. If you've seen it before, I'm probably boring you a bit. I want to point out a connection to another well-known problem, the longest common subsequence problem. And in a way, if you look at this example or also the previous one, they have something in common, and there's a relation between the two. A good alignment tends to have a long common subsequence, namely this matching part. So in a vague sense, the optimal edit script gives you somehow the longest match, but it's not identical. It would be, so here's an example showing two strings. Okay, I've left out the gap symbols to make it stand out clearer. If you put the optimal alignment here, the edit distance would be the number of positions where you don't have this gap. So we have 1 here for this mismatch, then here's a mismatch. And here's another gap. So edit distance would be 8. But if you look at the longest common subsequence, it's actually 6 A's. But here the optimal alignment only puts 5 of these A's in correspondence. And if you look at the example for a minute, you see the problem is that we charge a replacement as cost 1, same as insertion and deletion. Whereas finding the longest common subsequence treats a replacement of a character as an insertion followed by a deletion, or the other way around. And so the costs kind of don't match exactly. If you were to only allow insert delete, then they're exactly the same. In the original edit distance, where we allow replacing a symbol by another at the same cost as an insertion and deletion, you cannot directly reduce it to longest common subsequence. Although in spirit, the two problems certainly are related. Okay, that's just as a piece of warning, if you like. In the next section, I want to briefly recap dynamic programming as an algorithm design paradigm. This will certainly have been part of some undergrad algorithms modules, so I'll go quick. But I think it's worth, first of all, I like the template that I'm going to show you. I think it's worth to reiterate that. And it also means for the other algorithms, we can stop at a certain level and leave out the automatic part. I think overall that makes the presentation still more concise and more enjoyable, maybe. The way I teach this, I say dynamic programming has these 6 steps. And I'll show you on the example of edit distance what I mean by these. We always have to define what subproblems are of the original problem. And we have to somehow solve the original problem as a special case of one of these subproblems. Then to make progress, we do a local brute force step, which usually means guessing one part of a solution, but then trying all possible guesses. And from these two things, we can formulate a recurrence for the cost of the solution, or the quality of the solution in general. The other 3 steps, then once you've done this and you understood the general recipe, are automatic. You can turn the recurrence into a simple implementation that just memorizes the values. You can also replace this by, in practice, certainly more efficient bottom-up table filling algorithm. And so far, these 2 algorithms just give you the quality of the best solution. If you want to compute the actual solution, that also is always a mechanic process called backtracing. That's essentially a meta algorithm. It's an algorithm that generates an algorithm. And that one finds you the best solution. Okay, so the first 3 steps are where the creativity and insight into the problem are needed. The rest is automatic. If you want to prove correctness, you do this at the level of the recursion. And you can also guess what the final time is. Here, like, that statement is true if you stick to the recipe precisely. We'll see some cases where you can modify then the algorithms by using, well, additional knowledge about the problem and kind of beat the trivial solution, the trivial algorithm that comes out of the recipe. But in general, the time you get from just following the cookie cutter approach, you get the number of subproblems times the time you need to find, to go through all the guesses in one of these subproblems. And that product gives you the overall time and oftentimes also the space. Let's see this in action. So for edit distance, assuming you've seen this before, maybe it doesn't come as a big surprise. If you have a prefix of the 2 sequences already aligned, so you've constructed a prefix of the alignment or edit script, then the question is, what will this next column look like? And really, there's only 3 options. It can either be one that looks at the next character in each of the 2 strings, or it could be an insertion, or it could be a deletion. And you have to pick the best out of these 3 options. And because the overall cost of an alignment is additive, we count the number of edit operations. Each of these contributes a certain amount. These always contribute 1. This contributes 1 if the 2 characters differ. In each case, you know what to do. So if you map this onto the 6 steps, what you do for the subproblems is take prefixes of the 2 strings. So any positions in the range of the strings give you a certain prefix, and these are all our subproblems. The guess, as I said, is what the last column of the alignment looks like. And I guess at this point, we can also say the original problem is for I equals n, m, and n equals j, j equals n. That solves our original problem because then I've taken the whole strings. And that gives us a recurrence for computing the edit distance that directly follows this subproblem structure. If one of the 2 indices is 0, so I'm taking nothing from one of the 2 strings, but there's some parts from the other string. Well, let's take the first one. If I take nothing from the second string, then I have to delete all the characters I'm taking from the first string. So if I is given, that's I deletions. And the symmetric case, if I don't take anything from the first string, I have to insert everything in the second string that I have. And in both cases, these are the number of operations. That's what we get as the cost. If we're not in these degenerate cases where I have no letters from one of the 2 strings, then I can always do the 3 options for the last step. This one says I'm taking one more character from the top string, but I leave the bottom one alone. So that's essentially like, shouldn't have drawn it like this, I guess. I'm deleting this character. In this case, I'm inserting one in the second string. That's why this is +1. This is for a deletion. This +1 is for an insertion. And the third case is I'm taking the last string of both characters, and then I get a cost of 1 if and only if they're different. So here again, this notation, I know it as Iverson bracket. That's how Don Knuth calls it. It's 1 if the condition is true and 0 otherwise. Let's say else. That's a concise way to write the recursion. These are the three options I have for the last column of the alignment, and I take the best. That's essentially all for the experts that know dynamic programming. The rest is completely mechanic. Let's go through it one time. At this level, we can already estimate what the final running time will be, because the number of subproblems is fixed now. That's all of those, n times m. That's an n. Sorry, which one was the n? Second. The time to optimize one specific subproblem is to find the minimum of these 3 items, so I can do that in constant time. So the overall time is n times m. N times m, hard to say clearly. In the trivial solution, that's also going to be my space requirement. And this is something we'll come back to. As I said, the first step is to just compute the costs, but we can do a backtrace to find the actual edit script. Just to illustrate the 4 steps very briefly, if you have this recursive equation, maybe you're concerned that it's costly to compute, but fear not. In the case where we have dynamic programming templates apply, we can always do this. We can always compute the recursion in a way that I don't have to recompute many things. If I memorize, I guess that's always possible. It's just the number of subproblems that's not too big. So the convenient way to do this is to have 2 mutually recursive functions, one that computes the actual value and another one that wraps it and says, oh, have I computed this subproblem? If yes, return the cached result. Otherwise, call the first function to compute the result. And for our concrete problem, this is what it looks like. The edit distance, that's the function that does the actual work, follows very closely the recurrence. You see the if statements exactly mirror the case distinction. Then I have to find this minimum of 3 things. So, okay, here I can just compute all 3 and take the minimum. It's really boring. And then the memoization part is to have a second function. That's the cached version of this computation. It starts, you have to, at some point, initialize this array of storing all the subproblem values. I'm doing this outside of this function and just assume this is already given. I somehow have a global variable storing this. And then in each step, I'm just going to check if my entry for this subproblem has already been computed. Then I return it. Otherwise, I first compute it. Again, the code is supposed to bore you to death because it's really showing we can stop at the level. As soon as we have this recurrence, we don't need to write down this code each time. It's very schematic and looks the same each time. This is often not the most efficient way to do it in practice because you're essentially doing unstructured random accesses to this 2-dimensional array. And for cache locality, that's not perfect. You often do slightly better if you just fill up all the entries in this table bottom up. That's replacing recursive functions with for loops, and so that's another benefit in practice. It's not a huge factor. I don't know if maybe a factor 2 is already optimistic for this. Okay, but still, because it's always possible, it's kind of nice to spell it out. The general form means you're building a DAG over all the subproblems. The edges in the DAG come from which subproblem uses which other subproblem in one of the guesses. And it has to be a DAG, otherwise your recurrence wouldn't work. You would cyclically depend on each other. So it has a topological order, and you can fill the entries following this topological order. In most cases, the subproblems are tuples of numbers, and then if you pick the right order to iterate through them, you follow a topological order. But I think it's nice to keep in mind that this is it works in full generality. Here, much simpler. The subproblems were just the prefixes. I'm taking the first I characters from the first string A, the first J characters from the second string B. And the way they depend on each other, you can visualize, if you think about this matrix that we compute, Dij. Make it a bit nicer. So that's the matrix Dij that we're filling. Say I'm writing this in the way mathematicians would write a matrix. So 0, 0 is the top left corner. Then if I'm computing a certain entry, the three subproblems that the subproblem ij depends on are the three subproblems around it, namely the one with I minus 1, j minus 1, the one with I and j minus 1, and the one with that's one, and the one with I minus 1 and j. They correspond to the insert, delete, and replace / match. So the edges in the graph would look like this, and that's all edges. So you can do many things in this case to get a topological order, but one way to do it is to go row by row, and then within each row by increasing columns. Okay, that's a long way to say lexicographic by I and j. And here's the code for doing that. Lexicographic is very nice because that maps into two nested for loops. We first go over the rows, and then we go over the columns. And the inner part, again, is literally copying in what the recurrence tells us. There's the two base cases where we take the special starting conditions. And then there's the other case where I couldn't be bothered to write out finding the minimum again, so I just left it as a formula. But you would probably want to substitute the code from the previous slide. The magic here from the topological order is that whenever I'm using array values here, the topological order ensures that they've been computed before I reach this point, so I'm never accessing null pointers. That's what we have to make sure of. Where here the initialization to infinity is irrelevant. Also, it's inconsistent with the comments. I think we don't use any of the values. We override every single one of them, so it doesn't even need to be initialized. Okay. I think so far is super easy. The last step is to compute the actual edit script. At this point, of course, you can analyze this algorithm, and it's two nested for loops. I'm sure you could do it. It's the same theta class as the memoized recursive function, but it tends to be faster, as I said, for essentially hardware reasons. But also the code, I guess, is a bit cleaner to write. The last step is now we know from this table, like, okay, I didn't emphasize this, we return at the end the bottom right corner of the matrix that contains the edit distance of the full strings. That gives us the cost of the overall, or it gives us the edit distance, or the cost of the optimal edit script, but not a script or the alignment itself. If we wanted to get that, we can do, again, a very schematic and automatic translation to get a backtracing algorithm. Yeah, I couldn't fully track down where the name backtracing comes from, and it's not even always consistent. Sometimes people call this backtracking, and this is horribly wrong. Like, don't do that. There's no backtracking happening. So I'm strict on backtracing because we just retrace our steps, the steps we took to compute the optimal value. You can always write this as a queued recursive function. And here's the example for our edit script. I'm assuming I already have all the edit distance values computed, and I'm assuming I keep all of them around. So here we really need this m times n space. Then I have a recursive function that starts with the outermost. So I would call it with m and n, the largest values for the entire subproblem. And the function is recursive. It has these conditions mirroring the recurrence. I regret I didn't have a small picture of the recurrence. We could have it, but maybe we can just puzzle it together again. If I is 0, I'm not taking any letters from the first string. So if I want to get to j letters from the second string, I have to insert them all. So I do that in my edit script. Similarly, if j is 0, then I have to get rid of all the I characters in the first string. So I'm doing that. The other cases that can happen is the last column in my alignment could be one of the three things. And here I have to distinguish if it's a match or a mismatch, because the operation in the edit script is different. First case is if my edit distance, if the minimum I'm computing for dij came from this option. So I'm going back in the j column, I'm getting rid of one character there. So that was an insertion. I kept the I characters from the first string, but I add one more in the second string. Well, if that was the best choice, that means the last operation in the edit script has to be the insertion of that last character in the second string. And how do I get the rest of the edit script? Well, recursively. So I've just called traceback on the subproblem that I'm picking the cost off here and append this one more operation at the end. And the rest is completely symmetric. If the minimum came from choosing a deletion, then I'm doing that with the appropriate traceback. If the last characters, if I'm not in either of these cases, that means I could have had this one. I had a match in the last case, then I'm doing a traceback for all those, but I don't do any operation for the match because the edit script does nothing. Otherwise, the only option that's left is I did not do an insertion or deletion, but it wasn't a match, then it was a mismatch. I'm adding the replacement operation. Okay. If instead of producing the edit script, you wanted to produce the alignment, that's an easy exercise. The same kind of code. You just replace these edit script operations with appropriate pairs of characters. We could do that as an exercise, but I feel that's even too boring to do it. The time for doing the backtrace, if I have all the values stored, is dominated by computing the values, so I can always do it almost for free. And so computing the optimal cost and the actual solution have the same theta class complexity. Questions up to this point? Do you guys need a break, or shall we do one more thing? Short break? Has anyone not seen edit distance? Oh, I know Finn did. I think it's maybe the easiest dynamic programming algorithm. So it's unlikely that you see dynamic programming without edit distance.
In front of the room.
Which one?
The map of mathematics.
Yeah, I know that one.
I find it a bit funny. Computer science is apparently applied mathematics, but fluid dynamics is pure mathematics. I find it very strange, but everybody has their own views on such things.
I guess you can just study the differential equation of fluid dynamics and call it pure, but I mean the name certainly tells you something different.
Yeah. In Germany, the people who do a mathematics degree do like dynamics.
What do you mean by dynamics?
I mean, I didn't study such a thing because I did a computer science degree in undergrad. But my mathematician friends were all doing, I don't know, Hamiltonian mechanics, the mathematics of it, and I found that very strange. To me, that seemed like a rather more physics.
Yep.
But in England, I think it's because of the whole Newton thing. They really think that mechanics is part of mathematics.
I'm not aware of that being so standard. There's usually the culture divide at some point between applied math and pure math.
Yeah.
And I think depending on the department, they might stress one or the other more. Where would you even put this? Is this applied or pure?
I'd say it's applied. No. If the question comes from studying a physical phenomenon, it's applied.
Yeah.
And if the question comes from, this is a funny problem to think about, good.
I think the question always just is how far do you have to go back? I mean, most of mathematics has some origins somewhere, but then
of course, we wouldn't study it.
Sometimes questions are motivated by questions, which were motivated by questions, which was originally motivated by physics.
By the way, how long is this lecture? I forgot. An hour and a half, 2 hours?
Yeah, an hour and a half. So usually it breaks somewhere in the middle. Today is kind of perfectly in the middle. Which is unusual because we started on time. It's not my style, but can either pretend we're getting better, or the whole audio nonsense is just helping.
Are there people watching online? Or will there be at some point?
One at least, yeah. So time to move on, I guess. What is much harder to say is whether people watch the recordings. As of course you get all sorts of statistics, but YouTube makes it really hard to. So it's very hard to see how many people actually watched most of the video from beginning to end. YouTube is not very good at showing you this. Yeah. They give you a retention time, but it's not the same. And of course, like asking in class is always nonsense because that's preaching the choir. Yeah, and I think one use case is to just check back on specific things at some point, which is not the same as replacing the live lecture with watching the recordings. Maybe I should do a proper survey at some point. Yeah. I mean, there's also the theory of delaying watching all the lectures until the week before the exam, which is, I guess, concerningly common from anecdotal evidence. It's probably not a smart strategy, or not one I would recommend. What can I say? Okay, time to leave undergrad territory a bit behind. The next few bits are at least motivated by more specific applications in biology. The techniques are still very basic. In this section, we'll look at global and local and semi-local alignments. And global is essentially what we've done so far. What I mean by that is we assumed that we've given exactly the regions we want to compare. Like in the initial hemoglobin example, there's these two subunits and there's a gene encoding exactly that protein. And the full protein, we expect to do very similar things. Then what we've done so far with edit distance roughly matches what we want to do. More often than that, actually, we're looking for similar regions, but we don't know exactly where they are. And we can slightly tweak our notion of edit scripts and of distances and still get the same thing. In particular, as edit scripts, you can include these operations that allow you to delete or insert at the end of the edit script for free. I wrote it here as ignore prefix or ignore suffix. You could have also said it's a free insertion or free deletion. And that's what then is called a local alignment. So you should think of it as you have some potentially long string, you have some potentially long other string, and then there's a region where they really have a lot in common. You might still have some mismatches and gaps and so on, but not too many overall. But you're really, as part of the algorithm, you're zooming in on the part that is most similar. That's the goal anyways. Now, if you think about how we defined edit distance, there's a problem because edit distance can only get worse if you add more of the string in. The more string you add, the more opportunities for having distances or having operations that you need to count. So for local alignments, you have to flip things around. Instead of minimizing a distance, you have to maximize some score. So you have to reward the matches explicitly. Otherwise, you'll always get the trivial solution, oh, just pick the empty string from both. They are very similar. That's an easy switch. It's essentially just putting the opposite sign there. And maybe while we're at it, we might as well give the matching parts an actual value so that we encourage the algorithm to pick the longest region that it can put in good alignment to the other string. So we'll maximize some abstract score. We'll come back to that a little bit. Usually here for the first part, let's just say we stick with unit costs or unit rewards. So a match contributes a +1, and the others, mismatches and gaps, maybe are penalties of -1. Now, the other thing we need to do if you think about our dynamic programming approach, is to incorporate these special operations. And they look like they're symmetric, but from the point of view of the algorithm, you handle them slightly differently. If you want to allow ignoring a prefix of the string, what that means is at any point in time in your dynamic programming matrix, you used to have these three options of where you can come from. And you pick the best of those three. Now for local alignments, there's a fourth option, which is just start from scratch and forget everything else. Which means there's a constant 0 that you can also use as your preceding state. But that's also all that is needed. This just means if I'm in a position ij, and I'm choosing the 0 option, that means I'm ignoring the first I characters of the first string and the first j characters of the second string, like simultaneously. So it outputs these two. So we're ignoring these. Ignore. ignore if you pick this, this 0 option. That's for ignoring prefixes. You could try to ignore suffixes in the same way. I think more convenient for the algorithm is just to say, at any position in the table, I'm allowed to stop, because that corresponds to the prefix up to I and j anyways. And so if I don't want to include the rest of the strings, just do nothing. Which means for the final step, we'll not just output the top right, the bottom right corner, but the best value over the entire matrix. It's another scan of the matrix, but hey. Of course, you could also keep track of the current maximum you've seen so far instead of scanning again, but that's not a big difference, so I'm ignoring this part. So the free suffix comes from stopping here. Which means we're ignoring these two. No, where are we going? I've already disabled all the scroll bars. I don't even know why this happens. Too small. So it's really a very simple change, and you can adapt the recurrence to reflect the change. The starting with 0 shows up in 3 places in the recurrence, because we kind of split this case up previously. We had these boundary cases where you could only do insertions or deletions, but this is exactly at the beginning of the alignment. So there, it makes no sense to pick the better option of starting from scratch or doing a bunch of deletions or insertions at the beginning. The optimum will always be just do the 0. And then you have this additional case in the regular interior of the matrix, where opposed to the 3 choices, we now have this 4th option. And I've flipped the signs around for the others. So this was a +1 for the distance. Now it's a -1 for the score because we do a mismatch. And similarly here, I get a +1 for a match and currently a 0 for a mismatch. I'm not sure if that was my intention. You could, of course, patch this to say minus the same thing with equal, not equal. So now it's always going to be one of the two. You can write it in different ways. The fear is if you pick the score wrong, then 0 will still be, it might still be better to just do nothing. But because we reward the matches, I think the problem is still sensible. And the final output is not the lowest corner, but you pick the maximum over all entries. What you can also see here is it's not too hard to play with the score. We'll briefly look at this more systematically in a minute. But if you wanted to somehow treat insertions and deletions differently from, say, mismatches, you could just put in different constants here. The algorithm couldn't care less. Okay. There's a twist on this variant, which I know as semi-local alignments. Also has this name fitting alignment. Sometimes you know that one string is much longer than the other, and you're just trying to find a kind of fuzzy match of substring A, of A as a substring in B. So you don't want to allow the prefix and suffix ignoring operations for B, but you do include them for A. This is much closer to finding fuzzy matches of a string in a longer string. So you can also try to solve it via methods there, but if you want the full generality of the scores, then you can use alignment algorithms. So what changes is that only some of the zeros are allowed. I'm no longer allowed in the middle of the matrix to just restart from 0. I can only do this when I'm at the beginning, when I've not used characters from I, then I can kind of insert the characters in B for free. I'm still gonna have to optimize at the end over not the entire matrix, but from the last column. So this is A, right? I want to keep all of A, but I'm fine to ignore any suffix of B. Sorry, the other way around. So this so I'm finding A in B, so I want to ignore suffixes of B. Looks right. I think the formula is okay. Okay, yeah. Any of these positions ignores a suffix of B, and that's what we want to do. And the rest of the dynamic programming recursion is as for the local alignments. So probably also with the tweak I've done here, I want to punish the mismatches somehow. The other option would be this, right? No, not like this. Okay, it's the same as for the other one. We're going to modify the scores in a second anyways. You can further play with this and do other variations and other combinations of which ones to ignore or which ones to include. I don't think these have special names. As I mentioned already, the algorithm is very flexible in terms of how you score different things. And that's indeed something for the biological application that is very interesting to have a closer look at. First of all, whenever in the algorithm we're recording a mismatch, we know exactly what character is mismatched. So we could easily say there's a special mismatch cost for replacing any character by some other character, depending on those two specific characters. And this is essentially what, at the very beginning, what happened in this amino acid alignment. There's some amino acids that are closer to each other. So you want to keep the cost of replacing one of these with one of a similar group lower. That's giving some small positive score. Not as much as a match, but still something. Whereas if you replace it with something that's very different, you might want to penalize that. In full generality, what the algorithm can support is any kind of score that gives every column in an alignment some value, so that the total score is the sum of the column values. And by column here, I mean the two characters on top of each other, not the columns in the matrix. Just to pick names for that, we can have a matrix that tells us if I'm replacing a certain character by a certain other character, what is the score for that? I'm insisting here that it's symmetric so that the whole alignment is symmetric to replacing A and B, swapping A and B. That tends to be what biologists use. I guess it's probably just a user interface confusion if it makes a difference. which order to put the strings in. From an evolutionary perspective, I could imagine it might make sense to say that one direction is more likely than the other. I think this is actually known that there are some cases like this. But here, for general alignment scores, the assumption is that it's symmetric. So you have the case where you either have a match or you have a mismatch. And then you have one other parameter for a gap, again, because it wants to be symmetric. It treats insertions and deletions the same. So these are both valued with G, usually negative, and the others are valued with whatever the matrix says. Okay, as before, the total score of an alignment is the sum over all individual columns. We've always used this implicitly, that the overall score is the sum of things. It used to be the number of operations, that's the sum of ones, and so on. So is all the same. It's all staying the same. Just phrasing it explicitly because we want to be as general as the algorithm easily allows. Okay, let's see some concrete example of what this looks like for proteins. And what people worked out is there's groups of proteins that are functionally similar. And here the matrix is sorted so that these are forming nice submatrices. And you see here, they actually get a positive score. So the largest numbers tend to be on the diagonal. But there's also some off-diagonal entries within these groups that are still considered a match or a positive score contribution. Whereas then for the others, you tend to have penalty effects, but they're not all the same. So the way this matrix comes about is from studying proteins that we know it's the same kind of protein in different species, and you compare them also across time. And you see how often one amino acid mutated into another. And essentially, the numbers that you see here are log odd scores. So you somehow compare how often do you see these amino acids in general without being paired, and how often do you see one turned into the other. And from on a logarithmic scale, you get these numbers. I guess they're then rounded because they're certainly not all integers. I'm just mentioning this as one example. Blossom matrices, there's a specific methodology of how to get these numbers from data. And that was what was used in the alignment I showed initially. That's producing this hemoglobin alignment if you use these numbers. Okay. The last bit that we want to generalize from the biological motivation is it doesn't really make sense that insertions and deletions are treated individually. This is a bit depending on the exact process of evolution or mutations that you're looking at. But there's several places where it's more likely that you have a consecutive insertion of a number of characters in one spot. So in a way, if you're already cracking the thing open and insert something, having a few extra insertions doesn't increase the cost in the same amount as having this first opening. So at the moment, we're treating a stretch of k characters consecutive insertion the same as some isolated single character insertions scattered through the string. And that's biologically not accurate. A simple way to fix that seems good enough, even though it's a crude approximation, I'm sure, is to say we have affine costs. So instead of just k times g for a gap of size k, we have some offset. And you should think of this one being much bigger. That's the kind of penalty to open an insertion round or open a gap. And then extending the gap is a much smaller marginal cost. This acute way that we came up with, and I haven't seen it somewhere formulated, but it's just a cute way to write this down. And in the alignments with columns, we had insertions like this before. Now, if you have a contiguous round of insertions, you could have the convention that the first one is marked as opening a gap and the others as continuing a gap. Then you can assign scores just based on these two characters, and that automatically gives you affine cost functions. That's just a hint that really dealing with these generalized affine gap costs is not a big deal, because all you need is one extra type of gap symbol. So you'll have one extra case in the dynamic programming table, but apart from that, not much happens to the algorithm. And you'll have a look at this in the exercises in a bit more detail. Okay. Questions up to this point? There's one more small twist that I want to show today, and that's modifying the problem for an often interesting special case, namely when you're only interested in alignments that have a small distance or a high score, or the statement that there is no good alignment. You just give up in this case. So the way to think about it in the alignment matrix, suppose we have the two strings A and B. So A on here, 0 up to M, B up here, 0 up to N. So the problem is, again, the same setup as the original edit distance problem. And let's focus on edit distance. Nothing really changes if you go to the more generalized scores that I've just shown. So a normal edit distance score, and say we do just a global alignment to keep things easy. And what would we like as output? There's another parameter. Make that in red. Plus a threshold K. And I'm only interested in the alignment or the exact distance value if it's at most K. If the edit distance of the two is at most K, and otherwise you're just saying that the distance is larger. And you don't have to specify anything further. You don't have to compute the exact distance and also not the alignment. I think it's natural that this is a requirement you could settle on. Because I said initially it's not a hard problem in the sense that it's polynomial time solvable. If your two strings are both around 1 million characters maybe, then the space already becomes a bit concerning. Like still doable-ish, but it starts to get concerning. If they become even bigger, it turns into a serious problem. But you may be fine to stop the whole process if the strings are really very different, because you're looking for these conserved regions, and if they are very different, there's just nothing to observe. Now, whether you need a global alignment of these very long strings, at this point, the details of the application might allow other workarounds. But let's suppose that we're in this simple scenario for pedagogical reasons, and we want to do this task of aborting when we can conclude that no good alignment is possible. As before, we have to fill this table conceptually. And we have to output this value down here conceptually. Now, if this value is bigger than k, then we could have saved us the effort and not do it. But let's suppose, I'm trying to draw it a bit more cleanly. Let's suppose it's not larger than k. Some value d less than k. Then there's certain things that cannot happen. For example, I think we haven't really presented alignments in this way. Let me add that at the bottom. If you have an alignment matrix that you filled out, during the traceback, you're always following one of the three steps, either top or left or diagonally. So the traceback reconstructs a path through the alignment matrix by following such a course. You can go up and left and so on. And it's always going to be some path that you follow, and the steps tell you exactly the edit operations, right? Now, a good path will have to stay somewhat, well, we'll have to mostly use diagonal steps that hopefully are matches, otherwise the score will be bad. And if you look at these paths, maybe this does fit here, if I'm scaling it down a bit. The path also has to kind of reach the top. You could say the last few bits it can jump over, but it has to somehow go from here to there. And if you remember how these things are defined, we're back to edit distance. These are increasing only. These are contributions that count the number of edit operations. So this is a +1 in each step or a +0, but nothing else. So if you ever reach a point where the entry is above k, this path was not going to lead to a good alignment anymore. In particular, for reaching this value d, you can have steps that leave d unchanged. These must be matches. You can have a bunch of steps where you go to d minus 1, so you had a mismatch here, or a insertion or deletion. But you cannot have an unbounded number of these, because they're bounded by d. And so in particular, what can never happen is that you walk very far away from the diagonal. Because if you did, I'm trying to draw this a little cleaner again. If you ever reach a point here, the difference, so if that's the main diagonal, oh god, this is, my straight lines aren't so straight, right? If this is 5 positions off the main diagonal, then one of the strings is 5 characters longer than the other. At the very least, you're going to have to insert these 5 characters. All right, so this is always going to be greater or equal than 5, because I'm doing 1, 2, 3, 4. Okay, forget about a 5, it's a 4. The point is, every step I'm doing off the main diagonal will mean I have to have at least a 4 here. It could be much worse. But the point for our application here is there's going to be a main diagonal. Let's pretend that the distance at the end, the difference between n and m is smaller than k. Otherwise, we can immediately say it's not possible. From this main diagonal, we can only go this size k to the left and right. And anything that lies outside of this can certainly not contribute a distance less than k at the end. Because to reach out here, this is already greater than k. It can only go up further when you continue, so it's never going to make it. What does that help? It means we can avoid filling the part of the table outside here. So all of this we don't have to compute. All of this we don't have to compute. The part that we do have to compute is of size 2 times k times the smaller of the strings. And if k is fairly small, that's substantially less than the n times m we started with. Okay, let me briefly summarize this and write down what we learned. So off the diagonal by d steps implies the distance, the entry there is greater or equal d. Which means we only need to consider this banded matrix, band of size 2k. And in a way, what's needed in the recurrence is to handle the boundary conditions that whenever you access options that would lie outside, you just don't do it. And what's a bit annoying to implement it is that you somehow want to shift the indices so that it fits into a rectangular range, a rectangular array. But that aside, I think the algorithm is very intuitive. It's not hard to modify the dynamic programming algorithm to take advantage of this banded width. But the code is more messy than helpful, I think. So whenever you access a subproblem where the difference between the prefix lengths is too big, you just ignore that option. It will never give you something better. And if you reach the bottom right that way, you'll fill it with some value. Now, if that value is less than k, you can trace back and you'll find an alignment. If the value is strictly larger k, then you don't have any guarantee because your actual alignment might not be the best that you can find. There could be something that briefly goes out here and then back in that's better than what you found. You're kind of forcefully restricting your alignment to stay close to the diagonal, which is okay if the final value is small, but is not okay in general. So if the final entry DNM, DMN, we don't get the correct distance. So this is not in general a replacement for the full matrix algorithm. If it's less than k, then we get the correct value, and we can trace back an alignment with that value because the optimal one cannot leave this banded matrix either. Okay. I could try to sketch the next idea, or we postpone it. It's kind of a departure. So this is that's the end of the kind of basic things. There's one more cool basic thing at the end, and I want to show some more sophisticated technique. But I won't be able to cover the entirety of it. Let's maybe postpone it till next week, and I just give you a kind of a sneak preview. The technique we're looking at is a general option to somehow slightly speed up things. To a certain extent, you can ask yourself, how much can you further speed up the computation of edit distance? We've seen with this assumption that you're only interested in the small values, you can do something. If you're interested in the general problem, any kind of strings, and you want the precise answer, how much can you improve upon this textbook undergrad algorithm that we've looked at, the dynamic programming one? We'll see one dimension to improve the space usage specifically, but keep the running time unchanged next time. Another option is to somehow play other tricks and try to speed up the general algorithm while not trying to avoid essentially filling the entire table. And what this exhaustive tabulation method is, can in essence be explained with a slightly different problem. Suppose we multiply Boolean matrices. Now, this is simultaneously a good and a bad example. It's a bad example because I'm well aware that there's more efficient matrix multiplication algorithms specifically. But let's ignore that. So all of these fancy divide and conquer variants and the tweaks and twists on those that try to get this exponent down. If I start with an n by n matrix A and an n by n matrix B, and I'm interested in their product, the basic method would need cubic time because it has to compute all these little products. Every entry in the result is a dot product of a row and a column. And in a stupid way, the best we can do is spend linear time for each entry. Now, again, ignore the divide and conquer methods to speed this up, which are essentially playing with that we're doing multiplications in a specific structure on numbers where I also have a subtraction. This is only possible because there's numbers I can compute with. You can modify the problem to not be standard multiplication, but some other combination of the entries, and then we don't know anything better, substantially better than this n cubed. But this is really, I don't want to get into this really. What I want to show you is something entirely different. And so please just forget about what you learned, that you can do this better. What can we do without any special knowledge about the multiplication, but about the fact that it's Boolean matrices, that the entries are really small? And Boolean is just the extreme case of that. What you can do is split the matrices into small submatrices. I'm going to assign these a size. So this is a t by t submatrix, and we'll pick t later. It's supposed to be square. So that means also this product matrix consists of these little squares. Let me be more smart and just copy it. And now, by virtue of how matrix multiplication works, if I'm computing this submatrix of the result, I'm essentially computing products of submatrices of the input and adding them up. Okay, element-wise addition. What does that help? Well, because these are Boolean matrices, I can make T small enough so that there's not too many different little matrices. There's only 2 to the T times T, 2 to the T squared, different little matrices. Just because they're all 0, 1s, that's the only option. Okay? So if I take 2 of these and take their product, there's also going to be just that squared many options for the output. So if I pick my T small enough, and I need a really tiny T, fair enough, but if I pick my T small enough, I can pre-compute not just all combinations of submatrices from A and B, but all thinkable products of tiny submatrices. And again, this looks bad, but if T is small enough, that's actually negligible. And then every operation here, every product is in constant time on a word RAM, because I can use the indirect address to jump into the lookup table by just taking the entries here and treating them as a number. Okay. I'll try to make this a bit more clear next time again. But the benefit is, instead of having an operation that used to be T cubed operations for doing these little products, we replace that by a constant operation. And so by choosing the right T, we can get the overall cost down, divided by some power of logarithm, log n. There's a power there that I forget. And that same trick we're going to try to apply to edit distance. Okay, that's for next time.